Microplastics are incredibly small fragments of plastic, typically defined as particles less than 5 millimeters in length. These tiny pollutants can be as small as 1 nanometer, making them virtually invisible to the naked eye, yet pervasive in our environment. They represent a growing concern for scientists and public health officials worldwide due to their widespread contamination. The issue is not just their size, but their persistence. Plastics are designed to be durable, and this durability means they break down extremely slowly, accumulating over time. This accumulation occurs in oceans, freshwater systems, soil, and even the air we breathe, creating a global challenge that is difficult to contain or reverse fully at this stage of understanding. These minuscule plastic particles originate from two primary sources. Secondary microplastics are formed from the degradation and fragmentation of larger plastic items. This includes items like plastic bags, bottles, fishing nets, and food packaging that break down over time due to exposure to sunlight, wind, and wave action. The other category is primary microplastics, which are intentionally manufactured at a small size. Examples include microbeads once commonly found in personal care products like facial scrubs and toothpastes, or plastic pellets, also known as nurdles, used in industrial manufacturing processes. These tiny particles easily enter waterways and the environment directly from their use or through industrial discharge, adding to the existing burden of plastic pollution. The ubiquity of microplastics means they are now found in almost every corner of the planet, from the deepest ocean trenches to their highest mountain peaks, including remote locations like the Antarctic. They have been identified in the air we breathe, the water we drink, and the food we consume. This widespread environmental contamination is alarming because it means constant exposure for wildlife and humans. Scientists have estimated that the world produces over 430 million tons of plastic annually, and a significant portion, around 19 million tons, leaks into the environment, contributing to this ever-growing problem of microplastic pollution, which is now a focus of intensive research. The consequences of this pervasive contamination are now becoming starkly clear, as studies have confirmed the presence of microplastics within the human body itself. Researchers have detected these particles in a concerning array of human organs and tissues. These include vital organs such as the lungs, liver, heart, and even the brain. Furthermore, microplastics have been identified in human blood, testicles, and the placenta, indicating that these particles are not just passing through our digestive system, but are capable of infiltrating deeper into our biological systems. This discovery raises significant questions about the long-term health implications of this internal plastic burden on our bodies today. The primary ways microplastics enter the human body are through ingestion and inhalation, reflecting their widespread presence in our daily environment. We consume them unknowingly through contaminated food and beverages. Seafood, particularly shellfish, is a well-documented source, as marine organisms often ingest plastic particles from polluted waters. However, microplastics have also been found in honey, tea, sugar, salt, beer, bottled water, and even fruits and vegetables, often due to contamination from plastic packaging or agricultural practices where sewage sludge containing microplastics is used as fertilizer. The average person is estimated to ingest a significant number of microplastic particles annually, potentially tens of thousands, through their diet alone, making food a major pathway. Inhalation is another significant route of exposure, as microplastics are present in the air both indoors and outdoors. These airborne particles can originate from the abrasion of synthetic textiles like clothing, the wear and tear of car tires, dust from plastic-containing building materials, and various industrial emissions. Once inhaled, these particles can travel into our respiratory system. Studies have shown that inhaled microplastics can reach the deepest parts of the lungs, such as the alveoli, where gas exchange occurs. The regular shedding of synthetic fibers from clothing and home furnishings contributes to indoor air pollution, meaning we are constantly breathing in these microscopic plastic fragments within our own homes and workplaces. Once inside the body, particularly the smaller particles known as nanoplastics, some microplastics possess the ability to cross biological barriers. For example, evidence suggests that certain particles can pass through the lining of the gut and enter the bloodstream. This absorption process is a critical step as it allows the plastics to move beyond the digestive tract and circulate throughout the body. The size and chemical properties of the particles likely influence their ability to be absorbed, with smaller nanoplastics being more prone to translocation across these protective barriers. This internal journey from the gut into the wider system is a key area of concern for researchers studying their effects. After entering the bloodstream, microplastics can be transported to various organs and tissues. Scientists have observed that these particles, once in circulation, can become coated with proteins and fats, which might affect how they interact with cells and where they ultimately accumulate. Real-time imaging studies in animal models such as mice have visually demonstrated microplastics moving through the brain and potentially even causing blockages in tiny blood vessels. This capacity for systemic distribution means that no organ is necessarily safe from potential exposure, and the particles could lodge themselves in sensitive areas, potentially leading to localized damage or inflammation over prolonged periods of exposure. The presence of microplastics within human tissues is concerning because laboratory studies have indicated they can exert toxic effects at cellular and molecular levels. These studies, often using cell cultures or animal models, suggest that microplastics can induce oxidative stress, a condition where there's an imbalance between free radicals and antioxidants in the body, leading to cellular damage. They can also trigger inflammation, which is the body's natural response to harm, but can become chronic and detrimental if persistently activated. While much of this research involves concentrations higher than currently measured in humans, the findings point towards potential mechanisms of harm that warrant further investigation in the context of the long-term, low-level exposure. Emerging epidemiological research is beginning to draw links between microplastic exposure and specific human diseases. A significant study reported in 2024 found an association between the presence of microplastics in arterial plaques and an increased likelihood of heart attack, stroke, or death in patients. Beyond cardiovascular issues, associations have also been suggested with conditions like inflammatory bowel disease and various forms of liver disease. While correlation does not equal causation, these findings are alarming and highlight the urgent need to understand the full spectrum of health impacts. There are also potential, though less established, links being explored with neurological diseases, metabolic issues like diabetes and certain cancers, notably colon and lung cancer. Reproductive health is another area where microplastics may pose a significant threat. Studies have found higher concentrations of microplastics in placentas from pregnancies that resulted in premature births, suggesting a possible link between plastic exposure and adverse birth outcomes. The placenta, a vital organ for fetal development, can accumulate these particles, potentially affecting nutrient transfer or hormonal balance. There are also growing concerns about impacts on fertility in both men and women. The detection of microplastics in testicular tissue, for instance, raises questions about their potential to disrupt sperm production or hormone regulation, though more research is critically needed to confirm these specific effects in human populations. Compounding the physical risks posed by the particles themselves is the chemical danger from substances embedded within or adsorbed onto plastics. Plastics are manufactured using a complex cocktail of chemicals, with estimates suggesting over 16,000 different chemicals are used in their production, many of which are known to be hazardous. Additives like phthalates and bisphenol A are notorious endocrine disruptors, meaning they can interfere with the body's hormonal systems. Other chemicals, such as per- and polyfluoroalkyl substances often found in plastics, are persistent and have been linked to various health problems, including an increased risk of cancer. These chemicals can leach out of the microplastics once inside the body, delivering a double dose of harm. One of the major hurdles in understanding the true impact of microplastics on human health lies in the inherent challenges of studying them. Detecting the smallest particles, particularly nanoplastics which are less than 100 nanometers in size, is technologically demanding and requires sophisticated analytical techniques. Furthermore, microplastics are not a uniform entity. They vary widely in size, shape, polymer type, and the array of chemical additives they contain. This heterogeneity makes it incredibly difficult to accurately measure exposure levels in human populations and to characterize the specific hazards associated with different types of particles. Standardized methods for detection and analysis are still being developed by the scientific community. Another significant challenge is extrapolating findings from laboratory studies to real-world human health impacts. Many in vitro cell-based and in vivo animal studies use concentrations of microplastics that are often much higher than those typically found in the environment or measured in human tissues. While these studies are crucial for understanding potential mechanisms of toxicity, it remains unclear how these effects translate to the lower chronic exposure levels experienced by the general population. There are substantial data gaps concerning the precise sources of human exposure, the relative contributions of different pathways like ingestion versus inhalation, and what levels of microplastics in the body should be considered a concern for human health, complicating risk assessment. Despite these challenges, regulatory bodies are beginning to take action. In September 2023, the European Commission adopted a significant restriction on microplastics, prohibiting the sale of intentionally added microplastics in products where they are released into the environment. This ban, effective from October 2023, targets items like non-degradable synthetic polymer microparticles, including loose plastic glitter. Furthermore, the European Union's Zero Pollution Action Plan has set an ambitious target to reduce the amount of microplastics released into the environment by 30% by the year 2030, compared to 2016 levels, signaling a strong commitment to tackling this pervasive issue. Globally, efforts are underway to address the broader problem of plastic pollution, which is the root cause of microplastic contamination. In 2022, a landmark resolution was passed at the UN Environment Assembly, where 175 nations agreed to negotiate a legally binding international treaty to end plastic pollution. Initiatives like the Global Plastic Action Partnership are expanding, with more countries joining to develop national action plans. Ongoing research projects, particularly in Europe, such as Plastic Heal and Aurora, are dedicated to investigating the health impacts of micro and nanoplastics, especially on early life development. These collective efforts, combining research, regulation, and international cooperation, are vital steps towards mitigating the invisible threat of microplastics.